chromatids.
Time to go.
Ember needs to stop alerting me. Alright. So, you see in both of them, it happened during G1, you see in one of them happened during G2. And so, we call this chromosomal and chromatid aberrations. A chromosome aberration has both chromatids with damage. A chromatid aberration has just a single chromatid with damage. If both have damage, that happened during G1. If only one has damage, that happened during G2. Right. Over here, we have some fancy words. Restitution, it just means fixes it. Okay, repair, restitution. Deletion, we lose a part of our chromosome. Broken end rearrangement. Now, this is a funny one. When you break your chromosome here, the broken ends reattach in weird ways. So, for example, we had a break here, we had a break here. Rather than putting itself back together like this, these two ends went loop and stuck themselves together into a ring. The chromosome has now become a circle. Circular chromosomes do not work. They do not function. So that is no good.
You say they reattach how in just circle?
Right, so you break one end, you break the other end, and then they just kind of loop into each other and stick together. We also have some other weird broken end rearrangements. So over here, two chromosomes ended up getting breaks. So both chromosomes have breaks, but instead of attaching back like this, the two big ones attach and the two small ones attach. And so now you've got a chromosome that actually has two centromeres. This is a dicentric chromosome because it's got two centers. And the other one ends up just as a fragment. So really weird stuff can happen when chromosomes get broken. Right? Remember, all of these are double strand breaks that we're talking about. Okay, so double strand breaks can lead to really weird things. And that's why they are so dangerous. Because these chromosomes will not function properly. Okay. It's even possible for pieces of chromosomes to switch with each other. So we mentioned this earlier, right? You've got the green chromosome, you've got the yellow chromosome, and it's actually possible if you have breaks for some of the green information to end up in the yellow, and some of the yellow information to end up in the green. Once again, this does not work. It's going to mess up your chromosomes, mess up your DNA, and mess up your cell. And so that kind of brings us back to this picture, all the way back from second semester, PRE1. So main chain scission, this is going to be our double strand breaks. Cross-linking, right? We had our intra and our inter, right? So this is with either between the same strand or between different strands of the DNA. And then point lesions, right? So this is going to be our single strand break. So double strand break, single strand break, and then this was going to be that covalent linkage, covalent cross-linking. Comes full circle, just like a ring chromatid. Target theory, we have talked about this before. The idea is that the cell has molecules vital to its function. If one of these targets gets hit, the cell is going to die. So these are going to be very, very important molecules to the cell. Proteins, for example, could be targets. The thing is that most targets have backups. You've got a lot of targets, right? Lots of proteins, lots of enzymes. There's only one molecule that is unique and irreplaceable, the one we've been talking about this entire time. Okay? DNA.
DNA.
That's right. So DNA is our real target, our master molecule. If DNA is damaged, the cell will die. So, instant death of the cell. That is one thing that can happen when a cell experiences radiation. Or it could have reproductive death, apoptosis, which is programmed cell death, mitotic or genetic death, and then mitotic delay interference with function.
So what actually would be programmed cell death?
Take a look at the slide.
Or we have some.
You'd have
I
slides
see.
for these, yes. Unfortunately, it does not look like we'll have time to go over these slides because the time is 1:29. So we'll call it wraps here. Tuesday.